Welcome back to our virtual classroom. In this video, our discussion will cover the structure of the plasma membrane, the surrounding of the cell. The membrane is made of several components. We'll talk about most of those as we go through our video today. The main portion, though, of any plasma membrane is a lipid bilayer with some proteins inserted into it, and they behave in an interesting way that's often described as a fluid mosaic. To understand this idea, I'm going to take you on an interesting mind tour, and where I'd like you to go is your own bathroom. Especially imagine a bathtub about half full of water, and we're going to do a few weird things here to this bathtub, so just stay with me on this mental picture. The bathtub's half full of water, and I want you to dump into the bathtub a few interesting things that you probably wouldn't normally put in your tub. The first is a laundry basket full of ping pong balls, and there are just enough ping pong balls to completely cover the surface of the water. Kind of a strange idea, but stay with me on this. Those ping pong balls represent the heads of phospholipids. We'll also throw in there a few toys, maybe a Power Ranger doll. Just for grins, we got a Power Ranger, and he's tall enough that his feet hit the bottom of the tub and his head sticks out the top. In addition, we're going to throw in a nice floating rubber duck that's floating just on the top surface of the water. We're also going to throw in a sponge, a nice water-soaked sponge that's sitting on the side of the tub. It kind of sinks to just below the surface of the water, and it hangs out there. So here's your tub, half full of water, covered in ping pong balls. Little Power Ranger toy is sticking out the top of the water. There's a duck floating on top of that, a little rubber duck, and a sponge floating just underneath the surface of the water. The last thing we're going to add is a child in a bathing suit. We got to keep things nice and appropriate here. Child in a bathing suit in the middle of the tub. And what this child begins to do is immediately, like any other sane child in a bathtub, begins to splash. But they're splashing in a very particular way. Their right hand is pushing forward, and their left hand is pushing back. So right hand forward, left hand back, right hand forward, left hand back, right hand forward, left hand back. They're kind of swirling the water around themselves. Can you picture this bathtub? Okay, if you see it, what I want you to do is focus on one single ping pong ball. That one single ping pong ball is swirling around this child. The little robot's kind of oozing along slowly. The rubber duck kind of floating along slowly. The sponge, if you can see it underneath the water, is floating along kind of slowly. They're all in motion. So this is a membrane that is fluid or flowing all the time. The ping pong balls, as I said, represent the phospholipids. The robot with his head sticking out represents a protein that goes all the way through the membrane. The rubber duck is a protein that is just sticking out the surface of the cell, and the sponge is a protein that's on the underside of the membrane. The child really doesn't belong in the cell. Get him out of the tub, put him to bed. Let's get on with this discussion. This membrane plays a very dynamic role in the cell's activity. It controls what comes in and what goes out of the cell. It also is the boundary that separates what's inside from what's outside. What's inside the cell we would describe as intracellular fluid, or ICF. The fluid outside the cell we would call extracellular fluid, or ECF. The fluid immediately around the cell is called interstitial fluid. This is an interesting word. We're going to be handling a lot of words as we learn the terminology for A&P as the semester goes on. The word interstitial means between the spaces, or the spaces between. So the interstitial fluid fills the spaces between the cells. A good way to think of that is to think of a brick wall. Are the bricks the only part of that brick wall? And the answer, pretty obviously, as you look at one, is no. There's mortar between the bricks. So the interstitial fluid is a little bit like the mortar between the bricks in a brick wall. It's the fluid between the cells. Now, there is, and there you're going to see these a lot. I know you're getting used to these things coming on screen like this. There's an animation for you to watch over in the study area on mastering in Chapter 3. You figured in the last chapter how to watch these, so please continue. We're going to see several more today. I'm not going to play these during the main video. They take up time, and there's no reason to use that time when you can go wake this up in mastering. In this image, we see a beautiful rendition in 3 dimensions of what a cell membrane or plasma membrane looks like. Focusing in the upper left-hand corner, we can see what we're doing in the main pictures. We're looking at a little slice out of the edge of this cell. And what I want to do is I want to start on the left-hand side and work our way up and around and talk about everything that's labeled here. Before I do too much, I want to show you the big purple membrane which sweeps across our picture. And I want to give you kind of an orientation here. If you were to look at a driving map, where you have maybe a map of Texas. If you hold that map in such a way that you can read the words on the map, they're all right side up, then you know automatically that the top of the map is north, the bottom of the map is south, the right side of the map is east, and the left side of the map is west. You have that orientation kind of in your mind already. When you look at a picture like the one we have on the screen right now, what I want you thinking is that above the membrane is outside the cell, and below the membrane is inside the cell. Just assume that anytime I show you a picture of membrane, and honestly, I'll show you pictures of membranes a lot as we go through A&P. With that rule in place, let's start right over here with cytoplasm. The cytoplasm is the watery interior of the cell. Look next at the phospholipid bilayer itself. It's actually a bilayer or a double layer of phospholipids. One layer faces in with the head down and the tail up, and the other side faces out with the head up and the tails down. So here's a nice little slice through the cell membrane, and I'll magnify it for you so you can see it a little better. And now let's start working our way around. We have a glycocalyx represented by the green thing sticking off of this cell. We've talked about this term before. Glyco means sugar, and the word calyx means a bit of a halo, and this is a halo of carbohydrates that stick off the surface of the cell. Some of them are attached to proteins, some of them are attached to lipids. More about those as we get into this chapter a little further. The next item here is the nonpolar tail of a phospholipid molecule. It is the double fatty acid tail of a phospholipid. On top of that, we see what is represented here as a little ball. That's the polar head of a phospholipid molecule. And again, this one single phospholipid in this membrane. The yellow thing right here is cholesterol. Now, you probably heard from several different sources through the last few years that dietarily, sometimes cholesterol is blamed as being a little bit evil. Honestly, truly, it's not. As I said in Chapter 2, cholesterol is a critical component for several physiological things in your body. It's what you make a lot of hormones from, but here we're seeing a key role of cholesterol as it's a part of, an integral, important part of the phospholipid membrane. Cholesterol helps to stabilize the membrane and make it more stable and less likely to being ruptured. The next item labeled is a glycolipid. Again, take the word apart. Glyco means sugar. Lipid, of course, refers to that kind of chemical. A glycolipid is a molecule that is part sugar, part fat. The next molecule that's labeled here is a glycoprotein. Again, take that word apart. Glycoprotein, it's a protein with a sugar molecule attached to it. Coming on down inside the cell, I want to point out several things that are labeled here. These two that are popping off the screen right now are peripheral proteins. Peripheral means at the edges. Peripheral proteins do not stick all the way through the membrane. They're either found on the inside or the outside of the membrane only. The next label here shows a filament of cytoskeleton. The cytoskeleton, represented in this picture by the blue strands, are protein fibers that help to hold the shape of the cell. Now, this picture, this image, shows an integral protein. An integral protein is a protein that penetrates all the way through the membrane. This particular integral protein, this one we would classify as a pore protein. A pore protein literally serves as a doorway to let some things come into or out of the cell. So there you go. That's a nice little quick tour of a phospholipid membrane. Let's talk a little about the things we saw in that picture. Membrane lipids, there were several different lipids we discussed. 75% of all the lipids in a membrane are phospholipid. That was the main structural part of that membrane we just looked at. It had polar heads, which are hydrophilic. The word hydrophilic means water-loving. Hydro means water, philic means to love. It also had fatty acid tails, and those are described as being hydrophobic. Hydrophobic refers to being water-hating. And again, a typical phospholipid has 2 fatty acid tails. 5% of the membrane lipids would be glycoproteins. We saw at least one of those. This is a lipid with a sugar or starch molecule attached to it. And those are useful sometimes for identifying the cell and for a few other things that we'll talk about in a few minutes. 20% of the membrane lipids are actually cholesterol. And again, cholesterol helps to stabilize the membrane. All right, on the next slide, we'll begin our exploration of membrane proteins. They do several things for the cells, including they let the cell communicate with the environment around it. This makes up about half of the total mass of the cell membrane. By the way, you'll hear me often interchange the terms plasma membrane and cell membrane. They're sort of the same thing, and I'll describe another day how these are a little different. It handles most, or proteins handle most of the cell's specialized functions. Some of them float freely in the membrane. Some of them are actually attached to structures inside the cell. And we'll talk about 2 types of membrane proteins. Integral proteins, as I said a little earlier, penetrate all the way through the cell membrane. Peripheral proteins, on the other hand, are found only on the inside or the outside. They can be either, but any one of them will tend to be where it is and it will stay there. Now, integral proteins first. These are firmly inserted into the membrane, and most of them penetrate all the way through the membrane. Now, the picture we saw earlier, by this definition, would have included a integral membrane, but most integral membranes do go all the way through, and as such, they're called transmembrane proteins. Typically, a protein inserted in the membrane has a region that's described as hydrophobic. That end would tend to be near the middle where the fatty acid tails are, and ends on this protein are also going to be hydrophilic. Those will be close to the phosphorus-containing heads on the membranes. These actually interact with the lipid tails on the phospholipid membrane and water that's both inside and outside the cell. What these proteins are commonly doing is serving as transport proteins, channels and carriers, sometimes as enzymes, sometimes as receptors. And yes, as you just noticed, there is a animation for you to watch on the mastering study area. Peripheral proteins are a little different than integrals. They are sometimes loosely attached to the integral proteins. They're also often attached to filaments on the inside of the cell. These do all sorts of jobs that are unique to individual proteins. Some of them are enzymes. Some of these are motor proteins to help move cells around and to help cells rearrange themselves as they're dividing, or help with muscle contraction, or in some cases they're used for cell-to-cell connections. Now, we do have, again, 2 more animations for you to watch over in the study area on mastering, so please go watch structural proteins and receptor proteins. So that's kind of the nice little tour of the bits and pieces we saw, mostly lipids or proteins with a few sugar molecules scattered around there. Now let's, over the next few slides, look at exactly what these membrane proteins are up to. There are really 6 main functions for membrane proteins. One thing they do is they transport things around. They are often used as receptors for signaling. They're used to attach cytoskeleton and extracellular matrix, and those are 3 of the 6 functions. More will show up over the next few pages. Let's look at transport first, then we'll look at receptor functions, then we'll look at attachment. So, transport. In this picture, we see, yes, there's another animation to watch, so just watch it when you get the chance. In this picture, we see this molecule here, which is sort of an open door, if you will. This is a pore protein that provides a channel, and in this example, the cause for items moving through that transport would be diffusion, simple diffusion, or facilitated diffusion, actually, which we talked about in our chemistry chapter. So in this particular picture, the red balls would be going onto the inside of the cell. Looks like there's a lot more on the outside. They have a passageway through, so diffusion would carry them through. Now, this picture, this section of the picture, shows us a pump protein. Looks like it's using ATP for energy, which means that this is probably going to move molecules against the concentration gradient. In this image, we see a protein serving as a receptor. Yes, another animation to watch. There is an active site on the top of this receptor to bind some sort of signaling molecule. This would include things like neurotransmitters, hormones, and other molecules that would signal a cell. Those signals binding cause all sorts of things. They may cause reactions inside the cell. They may cause the protein to change shape. They may do all sorts of interesting things. Next, let's take a look at attachment. Yes, another animation to watch. You're going to get tired of that, aren't you? Oh well, watch them. They're part of your class. In this picture, we see this protein right here that is anchoring the cell to things outside. Remember, if we're above the membrane, it's outside the cell. It's also sometimes used to anchor things to cytoskeletal fibers inside the cell. All right, that was 3 functions. Let's look at 3 more functions. Sometimes proteins are involved as enzymes. They literally are enzymes, and they cause chemical reactions to happen. Sometimes they're involved in intracellular joining more than one cell together. And sometimes they're involved in cell-to-cell recognition, almost like a name tag. So, yes, these are the last 3. Now, let's take a look over the next 3 pages at enzymatic activity, then intracellular joining, then cell-to-cell recognition. Here we see a protein or 2 in the membrane acting as enzymes. Looks like we've got 2 enzymes side by side, and in this particular case, they form kind of an assembly line. And if you look here, the first enzyme is going to grab a little green ball and turn it into a little red triangle. Now, of course, they're not really making green balls and red triangles in the cell. This is just representing. The second enzyme would grab hold of the red triangle and change it into a blue square. So this indicates that these take part in changing chemicals from one chemical to another to another. So yes, go watch the animation on enzymes over in the study area, and let's get on with this. Here we see 2 cell membranes right up against each other with membranes called CAMs, or cell adhesion molecules. For the best demonstration or mental idea of this, imagine a Western shirt that has not buttons but snaps on it. These are kind of like snap to fit buttons, and they're called CAMs, or cell adhesion molecules, and they help neighboring cells stick to each other. Now, in this picture, we see a protein involved in cell-to-cell recognition. The glycoprotein in the bottom cell is being recognized by the upper cell. The upper cell might be something like a white blood cell that's going to go around and check for foreign invaders to make sure things that are there belong there. Here's an interesting concept. There are structures in some cell membranes known as lipid rafts. About 20% of the outer membrane surface of some cells may be lipid rafts. These contain phospholipids. They contain a kind of lipids we've not discussed yet called a sphingolipid. They also contain cholesterol. They're more stable than the rest of the cell membrane, and they're great areas for making stable platforms for cell-to-cell communication. They're great for having membrane invaginations, little folded parts in the membrane. And when we get to muscles, you're going to see the first great example of lipid rafts. So keep this in mind for our muscle discussion, where a nerve cell touches a muscle cell and tells that muscle cell to contract. That area needs to be very stable and not move around. And the nerve is not going to move over the surface of the muscle. It's going to be actually attached to it. And it's typically attached in an area known as a lipid raft. On this page, we're going to focus a little bit on the glycocalyx. We saw it in that beautiful photograph or that beautiful image a little earlier. It is a sugar coating, if you will, over the surface of a cell. But the sugars in this case are certainly not there for sweetness and flavor. They're there to be functional pieces. They're often attached to either proteins or lipids, so we get glycolipids or glycoproteins, and each cell has its own unique patterns of sugars on the surface, and these generally are markers for cell-to-cell recognition. It lets your immune system recognize parts of you and find those as being different from something like a bacteria that's in you. So these literally are like name tags on the surface of a cell, and it's often said that your immune system is able to recognize self from non-self. So in effect, your glycocalyx is your cell's way of identifying yourself to your immune system. Cancer cells actually have the opportunity to change this all the time, and it's one way those cells can just kind of get absolutely out of control. Over the next few pages, we're going to look at cell junctions. Some cells in the body are floating all on their own, like a cork bobbing around in some water, but most aren't. The cells that would kind of float around on their own would include things like blood cells and sperm cells. They're not attached to anything at all. They're free floating or free swimming. But some cells, in fact, most of your cells are attached to each other. And cells are attached to each other a little bit like bricks in a wall in three main ways. So we're going to talk about three ways cells are attached to each other. Cells are bound one to another through tight junctions. Cells are bound one to another through desmosomes. And cells are bound one to another through gap junctions. And we'll look over, you guessed it, the next 3 or 4 pages at each of these in turn. So we'll talk about tight junctions first. A tight junction is exactly what its name implies. It's an attachment between cells that are impermeable. A beautiful way to understand this is to pick up a Ziploc bag and squeeze the top edges together. It's kind of a watertight seal, and tight junctions are exactly that. They're great at preventing fluids leaking between cells. These are really useful in places like, maybe your skin or the interior lining of your urinary bladder. You don't want those liquids leaking out. You want them to be contained. Looking at this picture, you can see 3 cells stuck together, actually parts of 5. There's a little edge on the left and a little edge on the right, but we see three whole cells in the middle of the picture. And if you look at this zoomed in area right here, well, we'll come down at the bottom and look at the zoomed in area. You can see that the edges sort of ripple together, again, very much like the edges of a Ziploc bag. And while these are not very strong connections, they are very leak-resistant connections. Imagine yourself holding a Ziploc bag that is full of water. If you turn that bag upside down and kind of gently shake it, nothing happens. But if you grab the edges, the lips, if you will, of that Ziploc and give it a good firm yank, what's going to happen? Well, it's going to rip right open, of course. So they're not really strong, but they are very leak resistant. And you'll see these connections along the edges where these cells in this picture join together, kind of here, here, and here. And those are designed to form a nice leak-resistant boundary between cells like we see here. Desmosomes are a little different matter. Desmosomes sometimes are functionally a little bit like a rivet or a spot weld, anchoring cells to each other. The heart muscle is a great place to find desmosomes. We'll see those when we begin looking at cell types muscle types, and in the next class you might take with me whenever we are actually studying heart. These are held together at spots called plaques with linker proteins between them, and keratin filaments go through them. Now, let me give you another mental example here, and you'll see this when we get to the next page, but I want to kind of describe this to you. Imagine having two pieces of cloth, maybe t-shirt material, and you lay these two pieces, these two squares of cloth, one on top of the other, and then kind of slide one over the other. Are they attached? No, you haven't done anything but lay them together. So what I want you to do is imagine having two squares of t-shirt cloth, and you're going to maybe lightly glue to the very center of each of those, like a little bullseye in the middle, a little Velcro pad, the carpet side on one and the hook side on the other, and now you stick them together. Are those two squares of t-shirt cloth now stuck together? Are they attached to each other? Yeah, they've got a little bit of Velcro holding them together. If you hold one in each hand and rip them apart, can you pull them apart? Actually, yeah, pretty easily. Let's stick those two pieces of Velcro back together, though. So now you've got your two t-shirt squares Velcroed together by one little dot in the middle. Pick up a needle and thread and sew with your needle and thread back and forth and back and forth and back and forth through that Velcro pad. You with me? All right. When you've gone back and forth maybe a dozen times, then grab hold of the two pieces of cloth that you Velcroed and then sewed together and tug on those. Can you tear them apart easily now? No, you can't, because the Velcro really reinforces, the Velcro attaches and the thread really reinforces. These are kinds of connections that reduce the possibility of cells tearing apart, and they're useful in places that are under stress in the body, like your skin or heart muscles. So that would look like this. Between cells, we have some pretty tough connections that hold cells in layers together. And again, they're called desmosomes. If you look at the bottom part of this picture, you can see the plaque protein, which is very much like the Velcro pad that I talked about. And you see the linker proteins called cadherins, which are very much like the thread sewing that I told you about. So again, this is very much a reinforcing or spot weld type connection between cells. The last cell connection type we'll talk about is a gap junction. This is a really interesting connection that is more important in some specific kinds of cells in the body. These are transmembrane proteins. These are pores. These are tubes, kind of like tunnels, that connect one cell to another. The proteins that form these are called connexons. Kind of a cool name. They're good at connecting cells. They're great for allowing ions to spread back and forth between cells. Ions in particular, but also sometimes simple sugars or other small molecules can go between, especially cardiac muscle, smooth muscle, and some nerve cells. That looks like this. We're back to our familiar picture, and we're focusing now on this area of the picture. Let's zoom in and look at the bottom. And what you're seeing here is that these gap junctions literally form tunnels or channels between neighboring cells. So ions can leak back and forth, and cells like heart muscle cells can tell each other when it's time to contract. All right, we are focusing in this video on the plasma membrane. The cell itself is surrounded by interstitial fluid, that fluid around the cell, and that fluid literally contains thousands of other substances like amino acids and sugars and fatty acids and vitamins and hormones and salts and waste products. And the plasma membrane itself allows the cell to decide what's allowed in and what's allowed out. I'll probably show you this same slide again on the next video, but we have now finished describing the plasma membrane, and we realize that its ultimate job is to keep things either in the cell that belong or out of the cell that don't. And in the next video, we'll look at exactly how that happens. So thank you for your time. Keep on studying. Don't forget to watch those animations over in the study area. This video's done.